Thank you for letting me here present you Meteor2, which is a new tool to perform microbial communities profiling on shotgun metagenomics data. So here you have an overview of the workflow. We basically take the metagenomic sample that we can align against one catalog, one microbial gene catalog that is actually structured into metagenomic species that is environment specific and that is also annotated. And then what you can see here is that from that one alignment file, we can get what I named the TFSP concept, is that from one you get the taxonomy, the functional, and the strain profiling. On the functional, you can see that you have the CASIMs, the KEGG, and the ARD annotations. And there is a fast mode in Meteor2 that uses basically a reduced set of signature genes that allows to perform the taxonomy and the strain profiling. So we benchmarked Meteor2 against all these state-of-the-art tools. Basically, we simulated some metagenomic sample for mouse and human microbiome based on magnified genomes. And we selected species that are represented in the database of all the selected tools. So here you have the taxonomic KL benchmark, especially the species detection with a F1 score. And you can see that Meteor2 is the best performing tool. Then the second one is Meteor2 Fast. And then we have Metaflan and SILF and Motu3 for the best approach. For the richness estimation, you can see that again, Meteor2 and Meteor2 Fast and Metaflan were the best performing approach. And for the species abundance, so this time we calculated the Bray-Curtis similarity compared to how gold standard abundance. And you can see again that Meteor2, Meteor2 Fast, and then Metaflan SILF were the best approach. So if we take a deeper look of what is going on, so here we have 10 profiles for the three best profilers. On the y-axis, you can see the relative abundance predicted by the tool. And on the x-axis, you have the gold standard. And what you can see that things are happening at the low for low abundant species predictions. Meteor2 is quite accurate at for this area, but you can detect a systematic pattern for Metaflan that underestimate the low abundant species and SILF that tend to overestimate them. And you can see at the bottom that Metaflan and SILF have false negatives. This has a consequence, several impacts, especially when you take shallow sequencing. So here we reduce the depth from 10 million to 5 and 1 million. And you can see that the difference of performance is increased between Meteor2 and Metaflan 4 and SILF by 45%. So now I move on to the functional benchmark. So here the task is basically to find the UniRef profile at the species level from our gold standard. And compared to human 3, you can see that Meteor2 is the best for the F1 score in detecting the functional content. And when we look at the sample level for the abundance estimation of this UniRef, you can see that Meteor2 is at least better than human 3 by 35%. For the strain benchmark, we detected that Meteor2 tracks 9% and 19% more strains than StrainFlan, or human and mouse gut. And so basically all these tools give you mutation rates. And what we detected is that the mutation rates detected by Meteor2 have a high correlation with the ANI of our gold standard genomes. So here you have the benchmark in terms of performance. So in time, Meteor2 Fast is actually the fastest approach available. And you can see that METEOR2, the complete implementation, is at the same level as the other available tools. And in terms of memory, we can see pretty much the same. METEOR2 is working quite fine. So we had an application case based on fecal microbiota transplantation. We took 71 samples profiled with METEOR2 against the human gut microbial gene catalog. And you can see here the application of the TFSP concept at one microbial species level. Basically, we get for Bacteroides stercoris its abundance. Then we manage to clusterize the cohort according to the strain, and we can associate to this data the functional profiles that allow us, for instance, to identify partial auxotrophies that are here that match quite well with the donor. So in conclusion, METEOR2 offers a complete toolbox for the metagenomic sample profiling. It's benchmarked. It's available for 10 ecosystems, easy to install and easy to use. There is a preprint available. And I would like to thank my co-authors and to thank you for your attention.
Thank you very much. Again, there's time for one question.
Yeah, so thank you for a very interesting talk. You showed some very impressive results compared to MicroBiome. But the principle is the same as MicroBiome, as I understand it. You're trying to find marker genes that are unique to a particular species, and then you're aligning against those. So why have you had an improvement?
It's because it's not marker genes, it's signature genes here. So basically, signature genes are genes that are quite often co-abundant in the sample. That's completely different of the marker genes that are just core genes in the genome.
Sorry, I'm not developing MicroBiome. It uses signature genes.
No, marker genes. It uses marker genes. It uses actually 200 genes that are common in all the genomes from one species. Okay. And in our case, it's genes that are co-abundant in several studies, several metagenomic studies.
Okay.
Okay, thanks again. I think we have to move forward.